Right, our next chemical to control the growth of microorganisms would be our surfactants, which include soaps and detergents. These are amphipathic molecules, and remember that word amphipathic means that the molecule has both a hydrophilic part and a hydrophobic part. So that's how it's going to be able to work its way into the cell membrane and disrupt it. So looking at the soaps, like what you're going to be using to wash your hands with, they're going to be dederming the skin. So that's going to be used to mechanically remove the microorganisms from the skin, so kind of scrub them off and wash them down the sink. But the soaps don't kill or inhibit the microbial growth. So, because remember, some of the microorganisms actually eat the soap. But we do have some detergents that can kill the microorganisms, like the quaternary ammonium compounds. These are cationic detergents that we use as disinfectants and antiseptics. How they got their name, if we look on the bottom right at the chemical structure, the ammonium refers to the nitrogen, and then the cation refers to the positive charge. So this is a cation because it's a positively charged ion. And then quaternary means 4. So you've got 4 different groups, chemical groups, off of this nitrogen there. So that's what quaternary is referring to. So quaternary ammonium compound is a really long name to say. So we just shorten this and we call them quats. So quats are usually what we call these. They're amphipathic, being a detergent, so they've got a hydrophilic part on one end of the molecule, and another end has a hydrophobic part. So they're able to get inside the plasma membrane and then disrupt it, and that's how they kill the microorganisms. So an example of a quaternary ammonium compound that I want you to be familiar with would be benzalkonium chloride. And so you can see an example of that over on the right here. Another example of a quat would be Cevicol, which is a mouthwash. And Lysol is another quat, so that's one of our household cleaners. And this is just a picture of what the quats are doing. And this one's just showing you Cevicol. And it's in blue, and you can see how it's kind of gotten into the plasma membrane and it's disrupting it. And so the plasma membrane doesn't stay together, and then things start leaking out of the cell, and then that will kill the microorganism. All right, our next chemical, we've got the bisbiguanides. And you don't need to know that name, but we've got the chlorhexidine. So that one is used a lot in medicine. And it's going to work the same way as the quats. So it's also going to disrupt the plasma membranes of the microorganisms. This one is only used as an antiseptic. So it's going to be used to scrub the surgeon's hands, scrub the patient's skin prior to surgery. You can also use this to clean out a wound. So you can buy this at the drugstore. You just dilute it with some water and you can clean out your wounds at home with this. So it's really good to use for that. The reason we like to use this to scrub the patient's skin before surgery is that it will stick to the skin cells and keep working and keep killing any microorganisms on the skin. So we say that it has residual activity. So that's why they like to use this so much. Alkylating agents are the next chemical group, and they include glutaraldehyde, O-thioaldehyde or OPA, formaldehyde, and ethylene oxide. Formaldehyde really isn't used in medicine, so I won't ask you about that one, so you can kind of cross that one out. So what these alkylating agents do is they're going to attach an alkyl group, which an example of an alkyl group would be a methyl group, so a CH3. So they'll attach those to things like proteins or DNA, and that will end up cross-linking things in the cell and make it so they can't do their jobs anymore, and then that can kill the microorganism. These alkylating agents can be pretty harsh, and so they're not going to be used as an antiseptic. They won't be used on living tissue at all. Glutaraldehyde and O-phthalate or OPA, they're going to be used as disinfectants, especially for disinfecting things like endoscopes. So they will place the endoscope in a bath of the glutaraldehyde or OPA and just let it soak in there, and that'll disinfect them before the next patient. The difference between these two, the glutaraldehyde really stinks and it's pretty irritating to breathe in. And then the OPA does not have an odor and it doesn't irritate you when you breathe it in. So that's pretty much the difference between them. So besides glutaraldehyde and OPA, ethylene oxide is another alkylating agent, and it's actually a gas that's used to sterilize heat-sensitive materials. So if you have plastics that can't go in the autoclave, you can use ethylene oxide to sterilize it instead. So ethylene oxide is not used by hospitals and clinics very often, so it's probably going to be used more in manufacturing. And that's because ethylene oxide is toxic. It has to be aired out really well before you would open up the ethylene oxide sterilizer. So it does take a couple hours to run a cycle and make sure it's the gas is all removed before you open the chamber. And if you do breathe in the gas, it's very irritating to breathe in. It can make you throw up, and it can also cause cancer. So it's not a very good one to be around. And if it's used in manufacturing, if you look on the package, it'll say sterile and then EO, which just stands for ethylene oxide. So it can be used to sterilize things like suture, surgery gloves, just anything that might melt in an autoclave. You could use ethylene oxide instead. And then here's just a smaller ethylene oxide sterilizer, and it just put your packs and things in there, and then the gas is just piped in there, and it takes a couple hours to run a cycle. It's just run at low temperatures, so it's not going to melt plastics. And then you have to make sure that all of the gas is removed, so it takes a while to make sure that everything's been aerated out before you open the door, because you don't want to be breathing in any of that ethylene oxide gas. Hydrogen peroxide is an oxidizing agent, so it works like the halogens do, like the iodine and chlorine and fluorine. So it'll go around and take electrons from things and create free radicals, which can steal electrons and damage things in the cells, and that's how it can kill things. Hydrogen peroxide is used just as a disinfectant, so remember that's going to get rid of the pathogens on inanimate objects. An example, it used to be used to disinfect contact lenses. It can still be used for that purpose, but it's not used very commonly for that anymore. You don't want to use it on tissues, and that's because it can actually kill cells. So it's kind of an old wives' tale that to use hydrogen peroxide to clean wounds, you actually don't want to use it for that. If you get hydrogen peroxide on your skin, it actually will bubble up, and then when you rinse it off, you'll see that your skin has turned white, and it's actually killed your skin cells. So you definitely don't want to put that into a wound, because you've already damaged tissue there from having the wound, and you don't want to put hydrogen peroxide in there and then cause more damage and kill more cells. So yes, it does kill the microbes in there, but you're also going to be killing some of your cells as well. So it's not a good one to use to clean wounds. So if you want to clean a wound out, go to the drugstore and get some iodine or chlorhexidine, and that would be much safer to use. You can also take the hydrogen peroxide and vaporize it and turn it into a gas. And that's very good to use to sterilize heat-sensitive materials. So if you have plastics, you can run that at a low temperature and you don't have to worry about melting plastics. So some hospitals might have some sterilizers that use vaporized hydrogen peroxide. And then we can also use vaporized hydrogen peroxide to pump into patients' rooms. And you can't be in there while it's in there, but that will actually kill any C. diff endospores. So if you're working in a hospital, you may see that used in a patient who's had C. diff, and that will get rid of any endospores that were in the room. And the nice thing about the vaporized hydrogen peroxide, it's not dangerous because it will break down into harmless, non-toxic byproducts, so water and oxygen. So it's not going to be toxic like the ethylene oxide is. So let's make a list of ways to sterilize things. This would be a good thing to know for the exam. And we know that things are sterile when there are no bacterial endospores left. So if we've killed the bacterial endospores, then we know that it's sterile. Okay, so the different ways that we can sterilize things. We talked about using an autoclave. But remember that you can't use that for things that don't do well with high heat or maybe the steam. So if you have plastics, an autoclave would not be a good choice. We also have dry heat. And remember that's using that oven, and that's manufacturers would use that. And then flaming, like in the microbiology lab, putting the inoculating loop through the Bunsen burner, also called incineration. So those would be dry heat. And then we can also use radiation to sterilize things. And remember there are two forms of radiation. We have the ionizing radiation. So remember that would be like the gamma rays, X-rays, electron beams. And then we have the non-ionizing radiation. And remember that would be the UV light. And we can also use filtration. Remember we could filter either liquids or gases. Remember we talked about you could filter like injectable drugs because you don't want to heat those up because you could destroy the drug. So during manufacture they would run them through a filter and then the drug would be sterile. And we could also use ethylene oxide. Remember that's a gas, and that would be good if you have a plastic that you needed to sterilize because that runs at a lower temperature. And then the last one we had is vaporized hydrogen peroxide. So you just turn the hydrogen peroxide into a gas, and that's also good for plastics because it runs at a low temperature and it won't melt the plastics. Okay, and this just is a nice summary of the chemicals that we've talked about, so you can review that. Okay, and then there are some biological control mechanisms for microorganisms, and this is something that they've been looking into and researching. So we do have some predators. So Delovibrio, these are really interesting. So Delovibrio is a gram-negative bacteria that actually preys on other gram-negative bacteria. So in this picture over here, the yellow bacterium, that's a Delovibrio. And it will come up to this other gram-negative bacteria and start making a hole into its cell wall, and it will actually burrow inside of there and then actually eat the bacteria from the inside out, and then will reproduce in there and then come out and then go find another gram-negative bacteria to do the same thing again. So that's something that's kind of interesting to treat gram-negative infections. And then we've mentioned before using bacteriophages. So these are viruses that just infect bacteria, and they're very specific to which bacterial species they will infect. So you'd have to get the correct bacteriophage for the right species of bacteria that you're trying to treat in order for it to work. So they would kill the bacteria. And then we have these bacteriocins, which are proteins that are produced by bacteria to kill off closely related species. So they're kind of similar to antibiotics, but not exactly the same, but kind of along the same idea. And we're looking into that because we do have a lot of antibiotic resistance. And so this is something new that we don't have resistance to yet that maybe we could use instead.